but we're no longer able to offer pizza, but thank you for showing up anyway, for being here for the science. I'm going to let Chris introduce himself since he's graciously offered to do so.
So I see some familiar faces. That's nice. Thanks for coming. My name is Chris Pennell. I'm an associate professor in lab medicine and pathology. My lab is over in the Masonic Cancer Research Building in the Minneapolis campus. And my interests are in cancer immunotherapy and tumor immunology. And then I also, and this is just a plug if you want to volunteer, I'm the associate director for the Cancer Center's Community Engagement and Outreach Program. So we try to engage Minnesotans in cancer education and stuff. So if you have an interest in going to the state fair or going out and speaking to civic groups, please let me know, and we'd be happy to line you up. So anyway, so that's the introduction. So what I'm going to talk about is on the title here. This is a picture of Emily Whitehead taken on her 5th birthday in May 2010. 2 weeks after this picture was taken, Emily was diagnosed with acute lymphoblastic B-cell leukemia. And if this was in the 1960s, she would have had about a 10% chance of long-term survival. But since it was 2010, the ensuing 50 years of research upped that chance of survival to about 85 or 90%. So although her parents were devastated by the diagnosis, the prognosis was good. So Emily went through the standard chemotherapy treatment regimen for her disease, and the good news was that her disease went into remission. The bad news was that the remission was short-lived. And in 2011, it came back with a vengeance, and it was resistant to the chemotherapy that had put it into remission. So she went through 2nd rounds of chemotherapy, high dose, with the idea of then undergoing a stem cell transplant after the high dose therapy, put her 2nd recurrence back into remission. And so her 2nd recurrence was put into remission, but she became so sick that she was not a good candidate for stem cell transplant, and they ran out of options. So in the end of 2011, she was just given a few weeks to live. She lives in Hershey, Pennsylvania. Her parents, during the interim, were looking for other options for her, and they came across a clinical trial that was being conducted by Carl June at the University of Pennsylvania in Philadelphia. So they enrolled Emily in that trial. And I'll come back to Emily later. But what I want to talk about before I do that is cancer in general and cancer immunotherapy. So the odds are not in our favor when it comes to cancer. So as I mentioned, I'm the associate director. I go out and give these talks, and so I've done some of the math, and it's just amazing to me. So the estimate is that the average adult makes about 300 billion new cells every day. And there are about 12.9 billion nucleotides per cell. So if you do the math, the body is copying 10 to the 21st nucleotides every day in the body. The fidelity of DNA polymerases is such that they make an error in about 1 every billion or 1 in every 10 billion copied nucleotides. So if you multiply 10 to the 21st times 10 to the minus 9th or minus 10th, you're coming up with like hundreds of billions of mutations that can occur every day. In one cell, they're spread out over the 300 billion. A lot of mutations that occur every day. If you want to look at this another way, you can calculate what the length of the DNA is that is made in every day in our body. So the amount of DNA, if you were to stretch it end to end from a given cell, is about 2 meters, 6 feet long. So you multiply 6 feet times 300 billion, convert it to miles. The body makes about 340 million miles of DNA every day in our body. And to put that in a different context, that's almost 2 round trips from the Earth to the sun. That's a lot of DNA that's being made every day in your body. Mistakes are going to happen when you're dealing with numbers this large. So cancer is inevitable. That's the bad news. And so how do these mutations lead to cancer? I realize that this is maybe rudimentary for some of you, but I think it's worthwhile putting it up here. Cancer basically results from the accumulation of mutations over time, and our body has evolved these barriers, which I've depicted as walls, to prevent malignant cell, to prevent a cell from becoming malignant as it accumulates these mutations over time. And in the lower right-hand corner, many of you probably recognize the hallmarks of cancer from Hanahan and Weinberg, and you can imagine that each of those walls represents a different hallmark that the cell has to overcome for it to become fully malignant. And so Bert Vogelstein, the originator of the Vogel graph, says that cancers are basically like clocks. They count the number of mutations over time. And if you acquire a certain number or certain key driver mutations, the cell can become malignant. And for that reason, cancers are more prevalent in older individuals than younger individuals, simply because we've been around longer and we've had more time to accumulate these driver mutations, cancer-causing mutations. So obviously Emily is down here, and childhood malignancies certainly occur, but they're at a much, much lower frequency than they are in older adults. And these are data from Minnesota, collated from 2007 through 2016. So we've been exposed to more carcinogens in our environment that can damage the DNA. The DNA has been replicated. The inherent error rate and the mutational rate leads to mutations becoming more prevalent as you get older. And so the bottom line is that in Minnesota and around the country, 1 out of every 2 men and 1 out of every 3 women will develop cancer in their lifetime. So if you don't develop cancer, somebody you know will. So that's why we care about it. So I'm not going to talk about prevention and all that stuff. I'm just going to go straight to therapy. So what do you do if you get malignancies, right? And cancer therapy has evolved, as I mentioned earlier. In Emily's case, pediatric leukemia has gone from basically a death sentence to something that 90–95% of kids can survive long term. That's all due to research, right? So cancer therapy has evolved over time. And originally cancer therapy focused on just killing rapidly dividing cells. So conventional therapy kills rapidly dividing cells. It doesn't distinguish between healthy cells that are rapidly dividing and malignant cells that are rapidly dividing. All rapidly dividing cells are killed. And the reason why rapidly dividing cells are preferentially killed by chemotherapy and radiation therapy is that 6 feet of DNA is packaged into a nucleus that's about 1/5000th of an inch in diameter. That's a heck of a packaging job, right? So if a cell is rapidly dividing, that DNA has to be opened up such that it's accessible to the polymerases to be copied. And when DNA is unraveled from its protective histones and other proteins, it's susceptible to being damaged by chemotherapy and radiation therapy. But that rapid division is not unique to malignant cells. So the next phase in cancer therapy evolution was tumor-specific therapies. An example here would be Herceptin, a monoclonal antibody specific for a growth factor receptor that's overexpressed on about 1/3 of breast cancers. So it's not going to work for all malignancies, it's going to work for some, but it's tumor-specific and you don't get many of the toxic side effects that you do with chemotherapy. The way the field is going now is it's zeroing in on this bullseye, which is patient-specific or personalized medicine. And there are 2 major approaches for this personalized medicine. One is a pharmacokinetic genetic combination, and the other is immunotherapy. And I'll talk about immunotherapy in detail. But briefly, the pharmacokinetic genomic approach is where your DNA from a healthy cell would be sequenced, DNA from your malignant cell would be sequenced. They'd be compared to one another to find out which mutations are unique to your malignancy. And then if a drug exists that can target that mutation that you have, and you can handle that drug with your metabolic processes, right, they'll give you that drug. So if there are 4 drugs for chronic myeloid leukemia, for example, and I can tolerate drugs A, B, and C, but not D because of my, the enzymes in my liver, I'm not going to get drug D. I'll try to go with A, B, or C. So it's a way to try to focus in on what drug will be properly metabolized by your body and what drug can attack that malignancy. So that's the pharmacology genetic combination approach. The other approach is to use immunotherapy. And as you all know, immunology is the best studied and it's the absolute best field in biology. It's incredibly important and exciting. It can be used for everything. So that's why we're focusing on immunotherapy. So, and I'm sure Bruce would agree with that. Good. Thank you, Bruce.
Oh, no, I'm not convinced.
So what is immunotherapy and why is it useful? It uses the immune system to treat a disease, right? And it's been a game changer for cancer because it's specific. So the immune system has evolved to be highly specific. Specificity is the holy grail of cancer therapy. You can kill a malignant cell and not any other cell, including the normal cell from which it was derived. You're going to reduce the non-specific toxicity, widen your therapeutic window, and get a better outcome. So specificity is key, and the immune system has evolved to be specific. It's potent. One cytotoxic T cell can kill thousands of tumor targets. It's long-lived. You develop a memory response that can persist for years, decades, your entire life. And the other key thing is that it's adaptable. So one of the hallmarks of cancer is genetic instability. So even though most malignancies are monoclonal in origin, they derive from a single cell, the billions of cells, the billions of progeny of that cell. So I'm sorry, I'm going to take a tangent here for a second and I'll come back to the main point. I give talks to students, and middle school students used to scare the heck out of me, but they've turned out to be my favorite group because they're not too cool to be on their phone all the time, and they're old enough to know something, right? So they scared me at first, but now I think they're pretty cool. So I was talking about how a cancer cell can divide. So I said, so one cancer cell can divide to give two daughter cells, and then those two daughter cells can divide to give 4 daughter cells. And this girl raises her hand. She goes, "Hey, wait a minute. How come they're daughter cells? Why can't they be son cells?" And I said, "Okay, fair enough." So now I call them progeny, just to get around that. I love that. That girl was great. So anyway, so the progeny of a monoclonal tumor are not homogeneous because of this mutation rate, which is increased in many malignancies because one of those driver genes may increase the likelihood or decrease the ability of the cell to repair mutations, right? So a tumor cell is like trying to hit a moving target, and the immune system can adapt to that. So these are the 4 characteristics that make immunotherapy particularly attractive for cancer. And in 2013, Adam Levine was named the sexiest man alive by People, and Science magazine in 2013 named cancer immunotherapy as the sexiest drug. So it's been, immunotherapy has come to the fore at the turn of this century, but a few years ago with checkpoint blockade and everything else, the field really recognized that this is the case. And so that's the good news. The bad news is that if you're trying to submit a grant to the NCI, you're out of luck because the NCI, of all of the institutes in the NIH, the NCI has had about 150% increase in applications since 2013, and the funding has gone down. So the pay line at the NCI is now like 5% or something. So the good news is that the field is working well. The bad news is that everybody's jumping on the bandwagon and trying to get in. So woe is me. But anyway, so this is why it's cool. So I'm actually going to get to data soon. So one key function of the immune system is called homeostasis. And so when the body is injured or has a disease or something happens, and homeostasis is perturbed, the immune system becomes activated, and in a healthy individual, the net result is a return to that homeostatic balance. One of the homeostatic functions of the immune system is something called immune surveillance, where nascent tumors are eradicated before you even know you have them. So you can think of that as maybe being the last wall in that cascade of walls I showed before. And so this is a highly detailed diagram of how this works. So the immune cells recognize some of those tumor-specific proteins that have arisen via mutation and can kill those now foreign cells, right? So by definition, a clinically apparent tumor has escaped immune recognition, and the way I think about this is that they've erected a barrier between themselves and the immune system, and such that the immune system cannot eradicate the tumor, and the tumor can then grow and proceed. And so this is what stymied the field for about a century. We knew, when I say we, it's the global we, right? The field knew for about 100 years that immunotherapy would be incredibly potent to treat cancer, but this barrier has prevented us from using it effectively. And it's only been within the last 20 years that we've understood what the bricks and the mortar are in this wall that tumors have erected to prevent the immune system from attacking them. And with that knowledge, we can overcome this barrier in basically one of 2 different ways. So the first way is called checkpoint blockade. Many of you have heard about this, anti-PD-1, CTLA-4, and there's a variety of other molecules that can be blocked. So the way I think about that is that these molecules basically reduce the height of the wall, such that now the tumor can be visible to the immune system and it can be eradicated. Jimmy Carter is 94 years old. He's still from 92. He's still building houses for Habitat for Humanity. The reason is that he was treated, his melanoma was treated with a checkpoint blockade therapy, an anti-PD-1, PD-L1 combination. That allowed his immune system to control the tumor. He's still building houses. The other approach is to basically increase the number of immune effector cells such that they are like a tsunami that can break over that wall and kill the tumor cells. So it's a mass action effect. You have more effector cells and molecules than you do suppressor cells and molecules. And so you just get this effect that breaks over the wall. And there are two ways that we can do that. One is to use DNA, or I mean not DNA, but cancer vaccines. So you identify what is unique about the tumor that you want the immune system to recognize. You link it up with an immune-stimulating cocktail. You put that into the patient, and then you hope that it stimulates those tumor-specific cells to sufficient numbers and activity such that it can crash over the wall. That strategy hasn't been incredibly effective, mainly because the patient is immunocompromised because of the disease and the treatment he or she has had. The other strategy is to basically take those those tumor-specific cells out of the person and amplify them to large numbers and put them back in. And so the poster child for this is called CAR, chimeric antigen receptor T-cell therapies. And what is done here is that instead of pulling out the cancer-specific cell from the repertoire that the person has, you can genetically engineer the person's own white blood cells to recognize a tumor antigen of interest. The reason I put this diminution of the cells up here is that this took the field about 10 years to realize that you can make large numbers of cells, and if you put them back into the person without partially lymphodepleting that person, the therapy is suboptimal. And the reason is that the cells that you're putting in have to compete with the cells that are already there for stromal cell contact, cytokines, and everything else. So if you do a partial reduction of the cells that are in the body, it also reduces the tumor cells, but it reduces the competitors for the cells that you're putting in. And as I said, it took about 10 years for the field to recognize that this was needed because it's counterintuitive. Why are you going to do a lymphodepletion strategy when you're trying to activate the immune system? And so now you put in billions of cells that are specific for the same antigen, and they can go and kill the tumor. So that's what I'm going to talk about now. So the strategy that we've used and many other people have used is called chimeric antigen receptor. As the name implies, it's a hybrid molecule that's put together genetically. So the extracellular domains contain ligands for your target antigen of interest. Typically, they're derived from an antibody. Single chain FB is just the variable regions of the heavy and the light chain of an antibody with the specificity you want that are linked together. And so now what you've done is you've taken away the native recognition unit of the T-cell receptor, which is restricted to a peptide down to an MHC molecule, and you put on an antibody that can be made to pretty much anything. So you've really broadened the number of target antigens that you can target, that you can recognize with this molecule. So then when this receptor engages its ligand, it has to deliver a signal to the T-cell that allows the T-cell to proliferate, become activated, and become cytolytic. And so a naive T-cell needs 3 signals to become fully activated, one through its T-cell receptor, one through a co-stimulatory molecule, and one through a cytokine receptor. Signals 1 and 2 through the T-cell receptor and a co-stimulatory molecule are typically bundled together in the cytoplasmic tail of this receptor. So when the extracellular domain engages its ligand, signals 1 and 2 are delivered to the T-cell. Signal 3 comes through a cytokine receptor, such as the IL-2 receptor. When the paper that first described this came out, I think it was in 99 or 97 by Eshar from Tel Aviv. It was in PNAS, and I remember reading it thinking, oh, this is pretty interesting. No practical applications, but pretty cool genetic engineering. And it's a boat I missed. So, yeah, not that I could have done anything about it, but I clearly remember reading it and thinking, oh, this is pretty cool, but it's got no practical applications. So I'm an idiot. So the way that this works is that you take out a person's cells. Typically, there's negative enrichment for T cells, although you can do it with NK cells and other cells. You activate the cells such that they're proliferating. You then put in your gene of interest via a variety of different techniques. We use retroviruses. You then expand them up to large numbers, billions of cells, partially lymphodeplete the patient, and put them back in. And the paradigm for this field is CD19 specific CAR T cells. And the reason is that CD19 is low-hanging fruit on the tumor antigen tree. It's a cell type specific antigen. It's only really expressed on B cells, cells in the B cell lineage. It's not expressed on any other cell type except for some rare dendritic cells. And so if you target a CD19 cell, you're going to target B cells but the patient will still be alive even though he or she would be immunocompromised. You're not wiping out lung cells or any other healthy cell type, right? So it's on a non-essential tissue. And, yeah, so these were the targets that were first used in mouse studies, and they proved to be effective, and that led to the clinical studies. And in 2017 and 2018, 2 CD19 specific CAR therapies were approved by the FDA. Chimera and Yescarta from 2 different groups. They both target CD19, and they cost a lot of money. So a single infusion of Novartis' product is close to $500,000. The advantage of this strategy is that the patient is alive. These are given to patients that have failed every other therapy like Emily. So end-stage patients with terminal diseases have been cured by these therapies. They're also called living drugs because these T cells persist for years in the patient. It's not entirely clear why, but the prevailing hypothesis is that the stem cells from which B cells are derived do not express CD19. So when a hematopoietic stem cell commits to the B cell lineage, at some point, at a specific point in the process, at pre-B cell differentiation stage, CD19 is turned on. So there's a constant source of antigen stimulation for these CAR T cells from newly arising pre-B cells. So the cells are constantly being stimulated and they persist in the patient for years and years. It's not known if this persistence of the CAR cells is required for the disease to remain in remission, but it's there. So it's not like you repeatedly have to get the drug, it's a one-shot drug, and it costs a lot of money. But I'm sure the cost, I shouldn't say I'm sure, but I would hope that the cost is going to come down over time. So these are just some of the results from the clinical trial, and I am going to get the data, my data in a minute. But the numbers were astounding because, again, these patients would have died, right? So now you're up to 70, 90% remission rates. The median duration of remission was not reached because the patients are still alive. And it's been long-term detected. 20 months was as long as they look. But like everything, there are some limitations, and the one that I've alluded to is the loss of healthy B cells. This results in B cell aplasia, which requires these patients to get immunoglobulin IVIG for the rest of their lives. There isn't enough IVIG in the world to treat all of the patients eligible for this therapy, so we have to come up with ways to get around it, and the field is. So that's a chronic morbidity. There are some acute problems, such as cytokine release syndrome and neurotoxicity. So what happens is that you're taking billions of these CD19 specific cells, you're putting them into a patient who has systemic B cells, systemic leukemic B cells, and what we're getting essentially is a body-wide, almost synchronous release of pro-inflammatory cytokines that's like a tsunami racing through the body, causing multi-organ damage. One of the organs that's damaged is the brain, and in a couple of cases, people have died from cerebral edema. There is some connection between these two, but some patients get cytokine release without neurotoxicity. Some patients get neurotoxicity without cytokine release. Typically, there's a connection where this occurs first and then this occurs later. But the etiology of these diseases is still not clear, which is what we're trying to focus on now. And the cytokine release is more easily treated than the neurotoxicity because preclinical and clinical studies have shown that IL-6 is one of the key mediators. So if you neutralize IL-6 or block the IL-6 receptor, you can really mitigate cytokine release syndromes. The neurotoxicity the etiology is totally unknown, so we have no way of dealing with that. And I was talking to a clinician a couple of weeks ago from Sloan Kettering who treats these patients, and he says that the morbidity of this neurotoxicity is something that really is, he thinks, the most profound problem because they can't release a patient from the hospital if the patient is disoriented or has other cognitive problems. And so even though the disease, they can tell within a couple of weeks that the disease is in remission and they would have released that patient otherwise, they can't because of these long-term chronic morbidities. So my work is focusing on, I'm just going to skip this because it's just my background. I think you've had enough. I've had enough of it. So we developed a mouse model to try to understand these neurotoxicities and come up with a way to get around it. So the model's got 3 components. The first is a transgenic mouse that expresses a human CD19 gene under the control of the endogenous promoters and enhancers, such that only the B cells in this mouse express human CD19. So that provides the sink of healthy cells. The second component are mouse T cells, so this is in the B6 model. Mouse T cells into which we put the human CD19 gene and then reporters like GFP and 5-luciferase. So we put these tumors into the mice. If we don't treat the mice, they die. The third component, and then we do the lymphodepletion to monitor or to mimic what's going on clinically. The third component is where we can take syngeneic or congenic B6 T cells and then retrovirally transduce them with a clinically validated construct. So the CAR construct that we put into these mice is being evaluated now by a colleague of ours at the University of Washington in Seattle. And they're all human components, so the nice thing about this model is you can take a construct that's being tested in a patient and put it into the mouse directly. So even though these are human-derived genes, the proteins they encode have enough homology with the mouse signaling proteins to allow these T cells, mouse T cells, to be signaled. And then the readouts are tumor efficacy. So we use bioluminescence imaging to non-invasively monitor tumor growth. How long do they survive? What are the toxicities? So weight clinical score, this is just a way to enumerate observations made about how the mice look. How long do they survive? Look at the tissue pathology and the transcript components. So that's the model. And what we focused on, and I'll show you some data with the tumor in a minute, but what we focused on is the toxicities that occur. So most of our studies are done in the absence of tumor. And what we're looking at is called on-target, off-tumor toxicity. So the target is CD19. It's expressed by the healthy B cells. So these T cells kill the healthy B cells. So you get this on-target, off-tumor. They're not tumor cells. And then we also can measure what's called off-target, off-tumor effects. And these are the ripple effects that these pro-inflammatory cytokines have on other tissues that do not express CD19. So by removing the tumor, we can look at on-target, off-tumor toxicity and off-tumor, off-target toxicity. And the mice that we use are hemizygous for the transgene. And the reason is that CD19 is kind of puts the brakes on B cell signaling. So you've got, in our mice, their normal mouse CD19, and then multiple copies of the human CD19. And if you look at the percentage of B cells in the blood of these mice, the homozygous expression of the transgene knocks the numbers down to about a quarter of what they were in wild-type mice. The hemizygous have numbers that are closer to wild-type. And so for this reason and a couple of others, all of our experiments are using hemizygous mice. I'll just skip that. This is the CAR construct that we put into the mice. It encodes a single-chain FE derived from a monoclonal antibody specific for CD19, so the variable light chain and the heavy chains are linked together by a short peptide that's flexible. Then there's a hinge region that allows flexibility on the cell surface linked to a transmembrane, and then signaling domains that deliver signal 2 and signal 1 to the T cell. There's a coronavirus T2A sequence put in here, and this basically is a protein-rich region. And so as the ribosome comes down and it's making the protein, as it makes this part of the protein, it kind of hiccups and then comes back down. So you can get two proteins from the same transcript. And then this is a truncated human epidermal growth factor receptor that is non-functional, but it expresses an epitope for which we have a monoclonal antibody. So we can use this as a way to detect transduction efficiency. And then also our negative controls lack the CAR construct, and the T cells are just transduced with this, such that we have the appropriate negative control. So that's our negative control. So what we did first was to see if these cells can kill, and they can. So this is a cytotoxicity assay done in vitro with varying numbers of effector and target cells. So the more CAR T cells you put in, the more cells are killed. These are just wild-type B cells from a wild-type B6 mouse, so they're not killed. B cells from our target mice are killed, and then just some other control. So the transduced cells kill in vitro. They differentiate and expand also in vivo. So if we inject these into mice and then pull them out 3 days later and look for the numbers of T cells in the spleen, we put in 1 million cells, and in the spleen we're finding 1 million cells. There are other cells in the body, but you can see how they've expanded. If we put 1 million control T cells into a mouse and look to see how many of them we can find, they're 2 orders of magnitude lower. So we know that the cells are expanding in the mice. And through activation markers and other cytokines, we know they're becoming activated. So they cause B cell aplasia. So if we look for the numbers of B cells in the spleen 3 days after putting in 1 million of these CAR cells, we can't find any in several mice, and in some they're knocked down. If you look for B cells in the control mice, you can find them definitely. If we look in the blood 55 days after transducing injecting these CAR T cells, we can't find any B cells in the blood. And yet in the controls they're there. So we're getting B cell aplasia, we're getting persistence. So all of these are mimicking what is seen clinically. So we were happy. Our model is mimicking what happens. So one of the first things that we did was to look to see, can these kill tumor cells? And the bottom line is that they can. So we injected 1 million firefly luciferase marked B cells into these mice. We let them grow for about a week. Then we gave the lymphodepleting dose of Cytoxan. This is a chemotherapeutic agent, so it knocks down the number of tumor cells as well. And then we put in the CAR cells the day after this, and then continued to monitor the mice. So these are the mice that received the CAR cells. In orange are the mice that received the same number of the control transduced cells, and then in black are the mice that didn't receive any T cells. So if you look for survival, you can see that the CAR-treated mice survived significantly longer than the other two groups. But in this experiment and some other experiments, some mice died earlier than the tumor controls. And so these had a sublethal tumor burden, and yet they were dying. So this made us think maybe they're dying from some toxicity, and they're not dying from the tumor because it's a sublethal dose. So that's when we started to look at some of these off-target effects and took the tumor out of the equation. So the rest of the slides I'm going to show you are from mice that did not receive a tumor. So we found that indeed they did induce toxicity. I don't know what the heck. Oh, sorry, I don't know why these are up here. They're my slides, I shouldn't have. So if you just focus on this left-hand column, the mice were pre-treated 1 day before adoptive cell transfer with 300 mg per kg of cyclophosphamide. And then what we did was we weighed them daily. And within 6 or 7 days, well, this was 5 days here, the mice that received 3 million CAR cells died, or we had to euthanize them. If they received a lower dose of 1 million cells, several of them died, but a couple of them were able to rebound, and then the others did just fine. This clinical score is just a way to quantitate appearance, activity, for texture. There are ways to look at toxicity in the mouse, and then survival. So these studies showed us that if we injected 3 million or more, I won't show you those data, CAR cells, the mice uniformly died within a week. So we wanted to know why that was. So what we did was we did one study where we looked at the integrity of the intestinal epithelium. So we gavaged mice with fluorescinated dextran. And then we bled the mice a few hours later and looked for the presence of this fluorescinated dextran in the serum of the mice. In control mice, we really couldn't detect any, which meant that the integrity of the gut was still there. But for the CAR-treated mice, we found significant levels of this phytylated dextran in the blood, which suggested that the epithelial barrier in the gut was compromised, and then pathologists confirmed that. And this is something that happens in patients. One of the organs that's damaged is the gut epithelium. We don't know if it's because of recognition of CD19 cells in Peyer's patches, but this is just one of the toxicities that we see. The other thing is that we did some nanostring analyses and found that components in immune-activated signaling pathways were upregulated. So gamma interferon response was up, alpha interferon, so the type 1 and type 2 interferons were up. The IL-6 pathway was up, which is consistent with what was reported clinically. So from these data, we then injected mice with a neutralizing IL-6 antibody and found that that completely ameliorated the toxicity. So the mice didn't lose weight, and they looked fine if we neutralized IL-6. So we know that IL-6 is a player in this. We don't know what cells are producing it. We think from our studies and other studies that it's a secondary effect, so the gamma interferon secreted by the activated CAR can activate endogenous myeloid cells that then produce the pathogenic IL-6. But these are some of the things that we're trying to iron out right now. So then if we look at pathology, and this is in the brain, we can find that there's extensive vasculitis. I'm not showing you the control for this, but these are PMNs and other leukocytes that are in around a blood vessel in the brain of one of these affected mice. There's also periventricular inflammation. So in some of the initial studies that we've done, it looks like this sort of this inflammation is more global. It doesn't look like it's regional specific. We don't know that for sure, but that's what we're going to be looking at. So there is inflammation in the brain. What I have to say, so this is again another slight tangent. I still do I still do work in the lab, partly because I don't have a lot of people, partly because I like to do it. And so I was down looking at the mice one day, and in this one cage, one mouse was racing around the cage like a maniac, and another one was in another cage was doing backflips repetitively. And I'd never seen mice do this before. So I talked to Harry Orr, who's a neuroscientist, and he said that that was reminiscent of obsessive compulsive disorders and suggested I look for neuropathy in these mice. So that was what kicked off our studies of the neurotoxicity. It was a conversation I had with Harry. But the mice were really, really weird. I never saw that before. And it's not consistent. Not every mouse does it. So there's some inconsistencies there. But it was eye-catching, I can say that. So we did an experiment where we wanted to see if the blood-brain barrier was compromised. So we injected Evans blue dye into mice. 4 hours later, we anesthetized the mice, perfused them, removed the brain, and then quantitated the amount of Evans dye in the brains of these mice. So these were mice that were, you can see that the numbers are small, so we're going to repeat this, but the mice treated with Cytoxan had a baseline level. The mice injected with controlled T cells were the same, but the mice injected with the CAR T cells had significantly greater Evans blue in the brain, which is consistent with that vasculitis that I showed you before. And then we started to do some confocal microscopy to find out what was going on in the brain. And on the left is an image of a brain from a mouse that received the CAR cells, and on the right is a control that received, it was actually there on my computer, EGFR T cells, so the control T cells. Blue is BAPI, green are microglial cells, and the magenta are the T cells that we put in there. And so you can see that the T cells are evident in the brains of these mice and not here. And we've done flow to show that the numbers are really increased here. And then you can see that there's a difference in the microglial populations in these two mice. And so the morphology is different. And when a microglial cell is the innate immune cell in the brain, it's a macrophage-like cell. And it's got processes that extend in an unactivated state. When it becomes activated due to the damage or inflammation, those processes retract and the cell becomes more phagocytic, and it starts to reshape neurons, and it does these things. So we think that these changes, it's much more intense than here. These are probably processes that are being stained with the antibody. This is more of the cell body, and so this is indicative of activation. And a colleague of mine, Maria Savanovic, who's an expert in microglial cells, has done some more analyses and has shown that in fact these cells are activated. So something's going on in the brain. And these are some of Maria's data where she's looked for a microglial cell specific marker in different regions of the brain and compared the CAR T cells to the controls. And it's evident that there are more microglia or more stained images here than there are down here. And these are unpublished data. The data I showed you before with the toxicity were published, but this is what we're working on right now. So there's microgliosis. And as a model for inflammation, Maria uses LPS, and she says that the amount of inflammation she sees in these mice is phenomenal compared to the LPS, which she thought was the gold standard before. So there's a lot going on. So what we want to do is we want to visualize this neurotoxicity and see what happens. So this is work that we're proposing to do right now. So these were images that were generated in the University Imaging Center. They have instruments that can clear whole tissues. So this is a brain of a mouse from a TBT tomato positive mouse. This is a clear brain of it. What they do is they take the brain or whatever organ you're interested in, put it into a hydrogel, which is the gel that is in like baby's diapers, right? Then they immobilize the brain in that, and then they put it in an electrophoretic apparatus with some detergents in there, and basically they remove all of the lipids and the opaque molecules that prevent you from seeing through the brain. And then with that, you can image using some techniques called this, this is a ribbon scanning confocal, but they just got a new instrument about 3 weeks ago that will allow you to image your entire brain in about 4 hours. And it's the only instrument in the world right now. So this is a 3D image that was generated from this clear brain. And there's something called the Allen Mouse Brain Atlas. There's an Allen Human Brain Atlas. And that atlas has defined coordinates that allow you to identify what region in the brain you're looking at. So you can correlate this with that. So in combination with Mark Sanders in the Imaging Center and Thomas Pengo, who's an informatics expert, we're going to try to find out where the car cells go, what do the microglial cells look like when they're in the neighborhood, and try to figure out what's going on. The other way to visualize neurotoxicity is using two-photon confocal microscopy. And this is a collaboration we just started with Eric Newman in the Department of Neuroscience. And what Eric and his colleagues can do is that they can remove the skull of a mouse and put on what's called a cranial window. So they remove part of the skull and replace it with a glass window. And then you can anesthetize the mouse, and through that window, use multiphoton confocal microscopy to eliminate the tissue and see what's going on in there. So this is a CAR T cell that we had put into the mouse 3 days before. And this is a blood vessel. So it's near that material. And then this is one that's in a capillary. And then they can also make movies. And so this is a movie. It's a 3D, it's a Z-stack image, so you can actually detect these cells. And the point of this was just to show that we could find these cells in the mouse.
Are they always by blood vessels, or are they all throughout?
No. So sometimes they're in them, sometimes near them, sometimes they're in the parenchyma. And so what we're interested in is that whole process. So from autopsies of patients that have had neuropathies, they've identified activated vascular endothelial cells with certain integrins are upregulated. So it's thought that the blood-brain barrier is compromised, the integrity is compromised, there's activation, which allows some of these cells to extravasate more easily. And then once they get into the brain, they can continue to secrete. So let's skip this. So this is Emily. This picture was taken this past May. So she's the first pediatric patient treated with CD19 specific CAR T cells. Carl June said that if she had died, the whole field would have died, because in pediatric cancer clinical trials, if the first patient dies, nobody's going to want to put their child and have their child be number 2, right? So the fact that she survived really allowed the field to go forward. Obviously, her family is allowing the field to use her picture and her story to try to promote interest in it. But she's disease cancer-free, 7 years now, so she's 14 years old, so there's a happy ending for this. So we're trying to use our mouse model to summarize, to really focus on the neurotoxicity and the neuropathy aspect of this, the side effect of this therapy. And I think with the colleagues we have here, we've got a lot of potential. So the mouse model was developed in collaboration with Bruce Blazer, Mark Osborne, makes the retroviruses for us. Eric and Amy's a graduate student in Eric's lab, so they're doing the multiphoton confocal microscopy. Maria's has her expertise in microglial cells and their biology. Brent is a pathologist who made those H&E slides. And Michael Jensen's our colleague at the University of Washington, who provided the car chemistry. So that's where we're at now, trying to figure out what's going on in the model, and there's some other data that are related but tangential, so I didn't want to express it. So thanks very much for your attention, and I'll answer any questions if I can.
With your mice that were exhibiting what you called like the obsessive compulsive disorder, essentially, how are you differentiating between like stereotypy and bored mouse? And how are you differentiating between like actual potential neural deficit?
I'm not.
Because bored mice will do the same thing. Like if they get bored enough, they'll go a little crazy.
Yeah. I don't know how to, we did one experiment where we were trying to look at behaviors. And so we looked at the behaviors of these mice in the open field, their ability to walk on a robo-rod and on a beam. And in those 3, we didn't see any differences between the control mice and the CAR-treated mice. There are other behavioral tests that we can do that focus more on the cerebellum or some other areas. And I don't know enough about that right now to tell you, but I do know, what I can tell you is that every mouse that we've looked at, that we've injected the CAR cells into, when we've looked at the brains, there's been some neuropathy.
Okay.
So I don't understand why there are some differences. And I've been working with mice for 30 years. I've never seen a mouse do a backflip other than a couple of these mice. And the other one that was racing around the cage. I've seen mice jump around and do stuff, but this guy was
like going crazy.
Going crazy.
Yeah.
It was, and it was, it was enough for me to talk.
Did he always
to
circle in the same direction?
He did. It was counterclockwise. But it was obviously it made an impression on me, or else I wouldn't be talking about it.
Yeah.
So,
I mean, something to try possibly would be to give them enrichment and see if the behavior stops.
yeah, I'm probably going to focus more on the biology than I am on the behavior, just because I don't know enough about it, and there's probably enough variability there. It's going to be hard to get funding. Yes.
Fascinating talk, actually. I guess I wonder, is there a peak in the neuroinflammation and does it resolve over time? Have you been able to answer that question? Or when it happens, it either persists or gets worse over time?
We know that there's so the answer is I haven't looked, but I think that there is a point of no return that is dose dependent, cell dose dependent. And that's why, that's probably one of the reasons why the mice are dying at days 5, 6, and 7. If we, one of the slides I showed, we injected 1 million rather than 3 million cells into the mice. About half the mice died, and the other half come back in terms of their recovery, their weight recovery. It would be interesting to look in the brains of those recovered mice to see what is going on and what the differences are. So I think that at least in terms of that gross measure of toxicity, there is a point where you can recover if you don't cause too much inflammation.
Have you noticed, are you using the same age mice or have you noticed?
Yeah. No, we try to use the same age mice. Sometimes we've used older mice and they seem to be more sensitive.
Yeah.
Normally we try to use them like in 3 months range.
Does IL-6 blockade prevent this?
Yeah, I don't know, so we're going to do that. Yeah. You would think it would, but I don't know. So, yeah, so we've got a model for how we think the whole thing works from the time you put the T cells in there. And I'm pretty sure that systemic inflammation is playing a role in this. And so my working model is that due to the compromise of the blood-brain barrier, systemic pro-inflammatory cytokines can access the CNS. But we know that there are literally tens of thousands of these CAR cells in the brains of the mice. And we know that the cells are activated. And I was wondering if they remained activated after they extravasated because of what they saw in the periphery, or if they were becoming activated in the brain itself. So we looked and there were B cells in the brains of these mice. So I think what is happening is that you're getting activation of the CAR cell once it makes it into the brain via recognition of the CD19 positive B cell in the brain that's causing a more focal release of pro-inflammatory cytokines. So I'm thinking that that may be more of a driver than the leaky systemic pro-inflammatory cytokines, but we don't know. So I've got trying to get money to look at that. Yes.
So very similar pathology happens in sepsis also, where
Yes.
IL-6 is like biomarker for the outcome of the patients. So in the mouse model though, when they tested the anti-IL-6 or anti-TNF-alpha, they found that therapeutic window is very small. So it also depends like if you give anti-TNF-alpha or irrigating therapy, the mouse don't survive. The mice don't survive this. Have you done, what time point did you give anti-IL-6 in your mouse model?
We gave them 1 day before we put the T cells in there, and then we gave it for continuously, daily for a week, and that's all we did. So we haven't done more in that way. So basically, we just tried to swap it out, and so I haven't done a more exact analysis. Yeah, Bruce.
Yeah, a couple questions. A quick one. I think you mentioned in the CD19 homozygous transgenic mouse that the B cell level was really low.
Yeah.
Why was that?
It's because CD19 can inhibit B cell differentiation. It's kind of like a it's not really PD-1, CTL1, but it has got phosphatase activity. So as a B cell receives maturation signals and activation signals in the bone marrow, it develops, right? And so the mouse CD19, when a B cell becomes activated, helps put the brakes on B cell activation, kind of like the way that CTLA4 does in a T cell. And so in our mice, they have 2 copies of the endogenous mouse gene. And then for the homozygous transgenic, it was put in as a back, and there are, I think, 9 to 14 tandem copies of the human CD19 gene in one chromosome. So you double that plus the mouse, and I think it's just putting severe brakes on the B cell development. I haven't looked in the bone marrow to quantitate where the deficiency arises. But in a wild type B6 mouse, for example, roughly 45% of peripheral blood cells are B cells. In the hemizygous, it's about 25%. In the homozygous, it's about 12%.
Yeah. And then, so in patients that get CAR T cells, I think when they do that transduction of those cells, they're hitting all T cells, so various subsets have the CAR receptor. I wasn't sure in your mice, was it one or multiple subsets? And if it's multiple, would that perhaps one of those subsets contribute more or less to the neurotoxicity?
Yeah, so I didn't describe that. So we don't do any subsetting. What we do is we do a negative T cell enrichment from the spleen, and then we just use that for total transduction. And so when we look initially, the ratios of CD4s to CD8s in the beginning are what you'd expect them to be, roughly 2 to 1. At the end of the transduction procedure, before we put them into the mice, it's about 10 to 1 CD8s to CD4s. And we haven't looked to see, we haven't done any further subsetting there. And we don't enrich those transduced cells from the population we put in. We measure the transduction efficiency and then inject the appropriate number of cells to get the 3 million that we want. And that's how it's done for the Chimera and the Yescarta. And that's why we're doing it that way. There are some clinical trials now where they try to put in a more defined cell product to try to get around some of these effects, and they're finding that there are cell type specific effects that can occur. And so, but that's what we're doing right now. Yeah.
Do you have any opinions on 4-1BB cars and CD28 cars?
Yeah, so the 4-1BB seem to work better because they allow the cell to become more persistent, and you don't get exhaustion as much as you do with the CD28s. So the CD28 is one of the type 2 signals that you can deliver. 4-1BB is another one. And some of the original constructs had CD28, but now pretty much everybody's going with 4-1BB. And every perturbation or combination that you can think of is being tried preclinically and clinically. So, but 4-1BB seems to be pretty good. ICOS is another one that seems to be good.
And even when in a gamma retroviral vector, that's what we're concerned about. This is a selfish question.
Yeah.
No, we were just reading some papers about 4-1BB when in a gamma retroviral vector, bad things can happen, like over-signaling. And that's what we have.
Yeah.
But thank you.
So I'm curious because there's 2 questions. One, you have a very humanized model,
Yeah.
which I think is really important for looking at this because there's other toxicities that are showing up. So I'm wondering if you can address how important it is for people if they're going to develop models like what you've done, how important it is to actually use a humanized system in the sense of having expression of human molecule in your target and what's your CAR T cell. And the other thing I'm wondering with the toxicity that's showing up, do you think that this is unique to the CD19 system in terms of this vascular permeabilization or leakiness, or are other toxicities going to show up that are each going to have to be addressed as we move down the line? So, we're not going to be out of a job, of course, if we have to do that, but I'm just curious, how generalized what you're seeing you think is also for the other CAR T systems in terms of toxicity.
Yeah, so the first question is, the more humanized you can make the mouse, the better. But if you have, it's nice that our system is humanized and it's immune competent, because you obviously have to have an immune competent system here. Other people have been trying to use, severely immunocompromised mice and then repopulate them with human stem cells. But there are all sorts of problems with that. So to answer your first question, I think it's very important that you have some human component in your model. And for us, it allows us to just take a construct that's being tested in you today. We can put it in a mouse directly. We don't have to mess around with it, right? So I think that's important. But it's always a balancing act, between how artificial you're going to make it. And then the second question is, I think that I think the systemic toxicity that results from systemic killing of healthy and leukemic B cells is really contributing to this toxicity. I think for a more localized killing event, like you would find with a solid tumor, for example, you're not going to get this sort of toxicity. I'm sure other things will pop up. There was a clinical trial that had to be halted where a Herceptin-derived CAR T cell was used for colorectal cancer because that the HER2 growth factor is overexpressed on a certain percentage of CRCs. The first two patients died from pulmonary problems. And it was found that in the pulmonary epithelium, that growth factor is expressed at very low levels. And prior to this, it was below the limits of detection. But upon pathology, they found that it was massively inflamed with these receptor-specific CAR T cells, and then further analysis showed that there was a subset of cells that expressed that. So one of the big problems with using mouse models is the inability to predict all of these toxicities because a mouse is a human. So there are other problems that have popped up. And so that caused the field pause. And one of the key questions is, what are you going to target? We've got the technology to make the cells and manipulate the cells, but the $64,000 question in the field, particularly for solid tumors, is what are you going to target?
And especially if you have something that's at very low expression levels, but then all of a sudden it's very vital that it's at low expression levels.
Yeah, exactly. So that's CD19 as the poster child because it's worked well. And so a lot of people are using it as a platform to try all of these other strategies that hopefully will be prove useful to them. So, all right, I won't keep you anymore. Thanks very much for your attention.
Yeah, I should open it. I'm looking at this window. What?